Good evening, everyone. Good evening, Renuka. Good evening, Khushi. Let's wait for everyone to join in and then we'll begin with our class, okay? So today we'll be discussing about what are proteins and how do a protein gets formed, the protein structure and all the other things. So depending upon the time, we'll try to cover up a lot of topics today, right? And also make sure that if you're new to our channel, do not forget to hit the subscribe button. In addition, also make sure that you download our application and explore all the courses that are running for you all. We have a number of courses running especially for the June 2023. So those of you who wish to, you can download the application and get yourself enrolled in the same. So let's begin with our class now. Good evening, Shana. Right, so talking about the latest courses running for you, so we have the practice sessions running for you all. So these are the PYQ practice sessions, helping you all for getting a practice of the previous year questions, because as the exam is near, majority of the students think of let's skip off the previous year questions, but no, you should not do that. You should make sure that at least especially during the end of the preparation, you should focus upon the PYQs, okay? So make sure that the PYQs are done on daily basis. In addition, we also have the revision booklets and the PYQ pointers available for you in a combo offer. So those of you who wish to, you can WhatsApp us on this particular number and get yourself these hard copy notes, okay? In addition, we have other courses also. Mock tests are also available. We have notes booklet, crash course is also there, and a full course is also running for you all, okay? In addition, we also have a complete guide course in which you can get all the lectures in a recorded format. So it is a complete, like a curated course for you all, right? Let's begin with our class now. So we'll be talking about what proteins are all about. So yesterday, if you all remember, yesterday we had a small discussion about what a protein is. Let's understand about it more, right? Talking about what are proteins, and for understanding proteins we need to understand about what are amino acids, right? So we'll have a separate class for the amino acids as well. Wait for that. For the time being, just understand what are proteins specifically. So proteins are actually the main functional unit in sense of for a particular cell to perform any functionality, for any cell to get any particular टास्क कंप्लीटेड, उस पर्टिकुलर सेल को प्रोटीन्स की रिक्वायरमेंट होगी. अगर हम सेंट्रल डॉग्मा की बात करें, द डीएनए गेट्स कन्वर्टेड इनटू द आरएनए, आरएनए गेट्स कन्वर्टेड इनटू द प्रोटीन, बिकॉज जो सेल है वो प्रोटीन की लैंग्वेज को बहुत इजीली डिकोड कर सकता है. इसीलिए प्रोटीन्स आर ऑफ अटमोस्ट इम्पोर्टेंस. अगर हम प्रोटीन्स के स्ट्रक्चर की बात करें या प्रोटीन की बेसिक यूनिट की बात करें, सो द बेसिक यूनिट ऑफ प्रोटीन्स आर द अमाइनो एसिड. जब दो अमाइनो एसिड्स साथ में एक बॉन्ड के थ्रू फॉर्म होंगे, साथ में एक बॉन्ड के थ्रू कनेक्ट होंगे, हम अमाइनो एसिड्स के बीच के बॉन्ड्स के साथ जब एक लीनियर चेन बनेगा, हम उसे प्रोटीन का नाम देंगे, राइट? व्हेन यू लुक इनटू द बॉन्ड ओवर हियर, सो द बॉन्ड इज गिवन द नेम ऑफ peptide bond, right? So peptide bond is formed between the two amino acids, and as a result of the peptide bond getting formed, हमारे दो amino acids एक प्रॉपर ओरिएंटेशन में ओरिएंट होंगे, एक साथ कनेक्ट होंगे, एंड एज अ रिजल्ट ऑफ द पेप्टाइड बॉन्ड गेटिंग फॉर्म्ड, हमारा अमाइनो एसिड्स, सारे अमाइनो एसिड्स एक लीनियर चेन में अरेंज होके प्रोटीन का फॉर्मेशन करेंगे, राइट? सो व्हेन यू लुक इनटू the just a second. So when you look into the various amino acids joined to each other in a linear manner in order to form the amino acids, in order to form the proteins, the bond formed here is a peptide bond, okay? How does a peptide bond gets formed? Let's understand about that now, राइट? अगर हम एक बेसिक यूनिट ऑफ प्रोटीन की बात करें, उसके स्ट्रक्चर की बात करें, सो लेट्स अंडरस्टैंड हाउ डज अ अमाइनो एसिड एक्चुअली लुक्स लाइक. Yes, so we have N, then H and H. On one hand you have C, then you have C double bond O, O and H. On one hand you have your R group, that is the variable or the side chain, and on one hand you have your main H group, right? So this is how the complete amino acid actually look like. This particular side is given the name of amino end, or the amino group is there. You have a carboxyl group, and when amino and carboxyl group are there, in the same manner you will have the second one also, and after there occurs a formation of the bond with the expulsion of one water molecule. We have the hydrogen bond getting, we have the protein peptide bond getting formed. How does this happen? Let's understand that also. Yeah, so on the first hand you have your, let's say, NH2, you have C, you have COOH, R group and H group. This one is given your C alpha. Same hand, in the same manner you have NH2, C, COOH, R and H over here as well. From here OH is released, from here one H is taken up. These two leads to the formation of water molecule. Water molecule gets released, and as a result of that you have the next, or the in the next step you see CONH over here. You have C double bond OH, you have R, you have H, then R and then H. So this is what your, this is how your actual uh, peptide bond looks like. Okay, so with this you have your peptide bond. Over here water molecule is expelled, and with the expulsion of water molecule, 1 ATP is also required in this process. Okay, so this is how the entire what do you say, the peptide bond is formed, right? It's very very important for you to understand this, remember this particular thing, because in the examinations students get confused, students tend to forget this basic thing. But please make sure that you remember that OH of the COOH gets removed off, and on the other hand you have the H from the amino group getting released, जिसकी वजह से एक वाटर मॉलिक्यूल का रिलीज होगा और एक एटीपी का यूजेज होता है. ओके, रिमेंबर दिस. इन वन पेप्टाइड बॉन्ड फॉर्मेशन, how many amino acids are getting used? So 2 amino acids are used in the peptide bond formation. One water is released. In addition to that you have one ATP being used up. Very very important for you to remember this. Okay, is it clear to everyone? Yes or no? Please let me know in the chat sections. If it is clear, then we'll move ahead with the further topics. Okay, please check whether this particular thing is clear or not. Quickly understand this, very very important. Time and again questions have been asked from this area, so remember this, remember about this. Then I'll give you a simple question to solve so that you can understand it. Then we'll proceed further with the other topics. Okay, please check whether it's clear or not. Clear everyone till here? Okay, let's see. So what are proteins all about? Let's look into the PPT also. Okay, so what are proteins? So proteins are the large biomolecules and the macromolecules that comprises one or more long chains of the amino acid residues, right? Proteins perform a number of functions, because proteins are the main functional unit for a cell to get the function performed. So it is involved in a diverse variety of the functions. For example, catalyzing the metabolic reactions, that is via the help of enzymes, right? Then DNA replication, responding to the stimuli, providing structures to the cell and the organisms, transporting the molecules from one location to other. So all these are the basic functions, the basic functionality that a protein performs. Now talking about the peptide bonds. So peptide bonds are the covalent bonds, remember about this. Then they exist between any two amino acids, leading to the formation of a peptide chain. How does it actually forms? I have already explained you. Then a partial double bond exists between the carbon or the nitrogen of the amide bond, which stabilizes the peptide bond, right? Then the nitrogen that is involved in the bond, it donates its lone pair to the carbonyl group, resulting in a resonance effect. Again, very important point to remember and understand. So make sure that you remember this particular thing as well. Then in the complete process of the peptide bond formation, we have the release of one water molecule. I have already explained you this. And peptide bond is a covalent bond, CONH. Then water is eliminated, so sometimes this particular bond is, or this particular formation process is also given the name of dehydration process. Okay, then a peptide bond is formed, is therefore the basis of most biological reactions. Forming the peptide bond is a requirement for all the life, and the process is very similar in all the forms of life. Okay, so peptide bond is making the basis of what? It is making the basis of all the biological reactions, right? Because obviously if the peptide bond will not be there, amino acid will not be able to connect together, they won't be able to bind together, and if there is no binding between the two amino acids, they will not be able to form the proteins, and then no functionality will be there. So what does this mean? That the complete peptide bond is, or the peptide bond is the main basis of the actual functionality or the actual biological reactions of the entire process, right? Remember this. So this is how the peptide bonds are actually formed. You have first amino acid, then the second one, H2O gets removed off, and as a result a peptide is, or the peptide bond is formed. And when two of the amino acids are joined together, we call it as a dipeptide, because two amino acids, or the two units of the two units are joined together via the help of a peptide bond, so we call it a dipeptide. When one more would be coming and attaching, we'll call it a tripeptide. So this is how the process continues. Is it clear to everyone? Yes or no? Just let me know in the chat sections. These things are very basics, but many students tend to skip these things, so I have added here for your revision purpose. Okay, now let's understand about the basis of the structure. Okay, let's talk about the structure of the amino acids, or structure of the proteins. So talking about the structure of the protein, initially when the amino acids join with each other via the help of the peptide bond, they form the first or the primary structure of protein. So we call the primary structure of protein as the linear chain, okay, in which there is no interlinking between the two linear chains that we have. So primary structure is linear in nature. From the primary structure, then you have the secondary structure getting formed. So how does secondary structure gets formed? From the primary structure, there starts to occur, or the linear chain which we have initially, this linear chain start to bind with each other in circular formats, or we can say they start to coil onto each other in such a manner that the second level is achieved. So you can find a number of hydrogen bonds in between the various amino acids linear chain. Imagine this is the first chain, and then this particular region is going over here, so it forms this structure. So there need to be some bonds in between the two in order to make that particular structure stabilized. So you have the secondary structure. Then from the secondary structures and before the tertiary structures, we have some structures given the name of super secondary structures, which are formed, that are again helping in the functionality of the organism. So over here you have the domains and the motifs. Okay, in the secondary structures we majorly have the alpha helix and the beta sheets being available, and in the super secondary structure domains and motifs are there, where we have the various alpha helix and the beta sheets getting joined together in a linear fashion. For example, alpha 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 or alpha beta alpha beta. There are a number of super secondary structures available which leads to the formation of the, or which helps in the functionality of the organisms, and then the super secondary structure coil onto each other to form the tertiary structure, and from the tertiary you have the final quaternary structure. In the quaternary structure we majorly have our molecular chaperones being there. For example, our heat shock protein, for example heat shock 60, heat shock protein family 60 and heat shock protein family 70. Okay, remember one more thing, that it is the secondary and the super secondary structures which are most functional. That is, if a cell is performing any functionality, it is the secondary structures and the super secondary structures which are helping in the complete of completing the main functions. Okay, so remember about this: primary, then secondary, then super secondary, then you have tertiary, and finally the quaternary one. Remember one more thing, for all of these structures to get formed, there need to be stabilization between the complete, or there need to be stability, and for stability bonds need to be there, and bonds will always be non-covalent only. You will not find covalent bonds in the complete secondary or the super secondary or any level of the protein structures. Reason for that is because the proteins are very dynamic in nature. There is no requirement of any high bond energy protein, or high bonds energy need not to be there, because the bonds need to be broken after few seconds. They might need to be bonded together after few seconds again. So point over here is कि यहाँ पर कभी भी हमें कोवलेंट बॉन्ड्स का फॉर्मेशन नहीं मिलेगा, बिकॉज हमारा प्रोटीन जो है इट हैव टू परफॉर्म अ लॉट ऑफ फंक्शंस. इट नीड टू मेक श्योर कि टेम्परेरली स्टेबिलिटी आए, function perform ho, and it can again and quickly without less expulsion of energy can convert into any other any other orientation also. So always remember, covalent bonds are not found in the protein structures, and this thing has been exactly the same thing has been asked in the examination. So please make sure that you remember this exception, and then you'll be able to easily eliminate this particular thing if in case any question is asked from you in the examination. Okay, just check whether everything is clear to you or not. If yes, then we'll understand other things. Clear, everyone? Very good, right? So this is regarding the structure of the proteins. I've not gotten into the depth of the topic because for that you need to we need more than 2 hours for completing. So if you wish to understand the complete topic in detail, then you can join our courses. We have our live courses, we have recorded courses available for you all, right? So this was a small topic I wanted to discuss with you. So we have discussed about what proteins are, what peptide bonds are, and how do we actually understand about the protein structures. Okay, so this was about it. Thank you for joining in, everyone. So if you wish to enroll in our practice sessions for the PYQs, you can download our application and get yourself enrolled. The link is in the description box. In addition, if you wish to place your orders for the revision booklets and for the PYQ pointers, you can directly WhatsApp on this number in order to place your orders. Okay, good luck everyone, take care, and let's meet tomorrow now.